Hello, everyone, and welcome to today's webinar titled, Want to Know If Your Cannabis is Safe? I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box, and if you can, address each question to the specific speaker and then click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on that Ask a Question box located on the far left of your screen. I now present today's speakers, Dr. Anthony Macherone, Senior Scientist and Cannabis Technical Lead at Agilent Technologies and Visiting Professor at Johns Hopkins University School of Medicine. Joining him is Timothy Bolduc, LCMS Application Scientist at Agilent Technologies. For a complete biography on Dr. Anthony Macherone and Timothy Bolduc, please visit the Biography tab at the top of your screen. Anthony, you may now begin your presentation. Welcome to both of you.
Well, thank you. And today we'd like to briefly cover some of the things that Agilent's been doing in the cannabis world for cannabis testing, regulatory testing. A lot of this material certainly applies to hemp as well. And my colleague Tim and I, we're going to present some material. I'm going to quickly go through just the top-level review of all the latest and greatest applications that Agilent has put out there. And Tim's going to focus more on the eMethods section and what that means for you, our customers. So before we get started, I have to make a disclaimer that Agilent products and solutions are intended for use where this is permitted by law, in quality control and safety testing laboratories. Okay. As a quick introduction, safety and quality testing necessitates a suite of analytical systems. They're chromatographic, mass spectrometry, real-time PCR. All these tools are needed to be successful when you're testing cannabis or hemp. And Agilent has developed a suite of these eMethods, which essentially assembles the application, the consumables, and everything that's needed to be successful, get you most of the way there, and just by loading this onto your system, and off you go. And it defines the instruments, the consumables, sample prep, data acquisition, data analysis, reporting tools, all of those things that you need for the method. There's simple client-side tools. Essentially, you download them. There's a little software package or application that you unzip it. It loads the methods where they need to go, loads the sequences where they need to go, puts everything in the right place, and you're pretty much ready to go from there with just a little bit of help from an Agilent consultant. The consumables kits, the cannabis consumables kits bundle all of them together. Cannabis kits, as we call them, very specific for those methods. Potency, the column, reagents, etc., are part of it. Pesticides, columns for both gas phase and liquid phase, and sample prep tools to do about 400 samples are ready to go right out of the box. So Tim will talk a little bit more about that. But it really supports, the Agilent portfolio supports the entire cannabis testing workflow, all the required applications, rugged platforms, HPLC, LC-MS, GC-MS, GC-MS, all of these tools can be implemented in the laboratory. Most of these operate on Mass Hunter, so one software for your entire laboratory. You don't have to have, every instrument has a different software that you have to learn. Primarily, it's all Mass Hunter. Fully developed workflows. And when I talk about a workflow, I mean from sample prep to reporting, everything you need from there to get the job done, get a billable out the door. And of course, our customer support and service. We've been heavily engaged in this market now for several years, and we have lots of experience. First, let's talk about our potency testing. Every country or region that requires legalization of cannabis or hemp requires potency, which is the determination of the psychoactive component of the product that you're analyzing. So delta 9 THC, its isomer delta 8 THC must be separated chromatographically. THCA, which is the acid phytocannabinoid, and a bunch of other cannabinoids are often regulated as well. These include cannabidiolic acid, cannabidiol, cannabinol, and cannabigerol. Those are the most commonly regulated cannabinoids in these various regions. Of course, most laboratories analyze for more, at least 11. Some labs do up to 17 or 18 or 19 of these particular cannabinoids. There's not a lot commercially available, so you very rarely see more than 19 or 20 cannabinoids in a particular method. And we first developed this on the 1220 platform, the Agilent 1220 platform, which is a self-contained HPLC, and you can see it up here in the corner. And this particular version was a 400 bar with a variable wavelength detector. But lots of our customers want to be more flexible, and so this method translates to the 1260 system, which is the mid-range modular. A lot of folks are probably more familiar with that LC system. And we built around this on-site consulting so the people that come and install the instrument can also train your laboratory. So within a few days, you're up and running. And of course, there's a cannabis and hemp potency kit that can be, part of the consumables that can go with this, and of course the eMethod, and the consulting service that could come with the 1220 or the 1260 LC. It comes with, it's 2 days of method set up now by a trained field service engineer, FSE, the instruction manual, the column, which is the pour shell column, the LC solvents, water and methanol, the modifiers, formic acid, everything that you need to get up and running, and of course trained in your laboratory. So next, let's talk about pesticides and mycotoxins, which is arguably the most challenging of all of the regulated tests. It's challenging because sample prep is critical, and we have a well-defined single-stream sample prep that is amenable to both gas phase triple quad and liquid phase triple quad. So one sample prep, and that can be split to both platforms if needed. Here we're showing the LC triple quad method developed by our colleague Pete Stone and others for the determination of pesticides and mycotoxins in flower, as defined by states where they have recreational, adult use recreational laws for cannabis use, California, Michigan, Nevada, Oregon, Washington in particular. So the target list includes compounds in those, but it can be translated to pretty much anywhere else in the U.S. And that couples nicely with this method here. So when you're looking at some of these states, Nevada, California, Canada, there are compounds in these lists that are not amenable to liquid phase. And you may have heard that, you can do qPCR in negative ionization mode and analyze these on one hardware platform. And there's no debate that you see a quantifiable peak when you go that route. The problem is it is not selective for certain compounds like pentachloronitrobenzene in particular. And there's a lot of other problematic compounds as well, captain, chlordane, chlorphenapyr, methylparathione in particular, that just don't work well with electrospray or even qPCR. So the most common approach you'll see in most of the laboratories, especially California and some of these other states that I mentioned, they will have both the LC triple and the GC triple. And so that previous method that I just showed you, which is on a Intuvo or 6470 LC mass spec system, partners with this on the Intuvo 7010 GC-MS system. So those problematic compounds, you can run as few as 5 on this system with this method, or you can do more. And we do have customers that are doing more than that. So you have overlap between the two platforms, which give you orthogonal power in terms of your data. But this, again, partners well with that previous application, and it's on the Intuvo platform. So it's a very nice small footprint, very popular, and A lot of good work went into this. We also looked at basically all the pesticides that are in the U.S. state list and in Canada, put them together, weeded out the duplicates, and came up with a target list of 100 compounds. And from that, from our own experience, from what was suggested for the analysis through organizations like AOAC, some other tools that we have at our disposal, it essentially said that of that list of 100 compounds, 27 of these are more amenable to gas phase. And so on our 8890, again using the 7010 GC triple quad mass spectrometer, we developed this application for 27 of the GC-Amenable pesticides from that list on this platform here. And again, this here is more pan-Americas. So it covers the GC-Amenable pesticides in Canada, the GC-MS-Amenable pesticides in the U.S. And so it really is something that can translate anywhere around these two countries. So it's very, very popular and, very robust. Next, of course, very specific to Canada, which has the largest list of 96 compounds with the lowest action levels or limits of quantitation, many of which are around 10 ppb in the various products that they've legalized. We have essentially a method that is both LC triple, GC triple, and of course that single stream sample prep that I described earlier, specifically for Canada. And of course this is an eMethod as well. So this is pretty much identical on the LC triple quad side and very, very closely matched to the GC triple quad work in the US, but this is specific to Canada. We next looked at, this is more recent work, through this recent months of chaos. I would say that we at Agilent have not been sitting on our hands and knees. We've been working. We've been getting things done. We're essential business, and we developed a very unique and novel residual solvents analysis for cannabinoid products. Now, you don't typically do residual solvents in plant material. This is more a process, oils, where things have been extracted with supercritical fluid extraction, those types of things. And you want to make sure that solvents that were used in that processing are not left over in the product. So things like benzene or methanol, ethanol, some of these common solvents that are used for these types of things. And so you'll hear people say, and in fact, very, very recently at the ASMS reboot, in the workshop, the cannabis workshop, they're talking about residual solvents, and one of the speakers said, it's essentially USP 467. Well, no, we learned very quickly it is not. You really have to look at these cannabinoid products as something different. USP 467 is very traditional pharma, single API, maybe two APIs for drug-like materials, drug-like products. If you're looking at some of these cannabinoid products, hemp extracts or cannabis extracts, there's just not one or two quote-unquote API in there, or active pharmaceutical ingredient, I don't want to call it, but active ingredient. There's many. There's lots of terpenes that may have properties. There's, of course, THC or CBD or both, and other cannabinoids. There's flavonoids. There's lots of other materials in these particular things. So we quickly learned that, this is not USP 467. And not only that, we really had to think about sample prep. It's not just weighing some material into a Headspace file, putting it onto the autosampler and analyzing it. If you really want good, robust results, you have to do a little sample prep. So this very, very detailed application note here, which is a Headspace application using the Intuvo with the 5977B mass spectrometer, single quad mass spectrometer, we developed specifically for cannabinoid products. And again, very early on when you read this, in fact, you can, in the abstract, pretty much we say, this is not USP 467. You have to look at it differently. And so this application does that, and very unique. We just completed liquid injection terpenes analysis on the Intuvo 9000 and the 5977B. And quite often you'll see in the cannabis testing world that Headspace has been the choice for this analysis. So, and Headspace has some advantages. You don't have matrix, you don't have some of these other things. There's a lot of disadvantages. You can't dilute. You lose a lot of the sesquiterpenes and the larger, higher boiling, later eluting terpenes, especially in the presence of high cannabinoid content. So it's kind of a problematic approach. And a lot of laboratories, especially over the past year and a half or so, have really been looking for a liquid injection approach. And Agilent is the first vendor out there, instrument vendor, to actually have an application for this particular. And again, very, very detailed. A lot of work went into this. And these applications, the residual solvents, the terpenes, the Intuvo Fast 5, as we call it, pesticides, or the 88-90-27 pesticides, all went through an internal vetting process where it's like a torture test. Multiple days, multiple calibration levels, everything run in replicates, and you end up with a data set of hundreds and hundreds of data points, and then you can look at that at intraday statistics to calculate things like the minimum detection limit, NDL, or LOD, limit of detection, or LOQ, limit of quantitation, range, linearity, accuracy, precision. And that's what we've been doing in all of the applications that Agilent's now, releasing, and you'll see this more and more as we move forward, some new things coming this year that are going to go through that vetting paradigm. So after we release them into the wild, as I like to say, we know that they're very robust. We also have qPCR, polymerase chain reaction, approach to analyzing for E. coli or Aspergillus species. These are very dangerous compounds. They're all regulated by the various states. In Canada, the products have to be free of these little buggies that can make us sick. And of course, our approach is the PCR. We have partnered with Medicinal Genomics, the manufacturer of the kits, multiplex kits, speciation type kits. For example, we now have, with our partnership with Medicinal Genomics, previously when you did a multiplex test, you looked for SPAC, E. coli, Aspergillus, and Aspergillus in particular, which has 4 different species that are regulated, you'd only get a positive Aspergillus. You wouldn't know what species it was. Now, within that same well, you're going to get speciation. Which one is it? Because some are more toxic than others. So that's our partnership with Medicinal Genomics using the ARIA MX qPCR platform. And as opposed to plating, there's a lot of advantages. And you can read the application note. But when you're plating, it requires hundreds and hundreds of plates to do these various tests for all the samples that you might have. And here you can do, in 20 minutes or so, all the tests you need in 96 well plates. So it's very quick, very efficient, of course, highly selective, because you're looking at specific parts of the genome for those particular bugs. We also have ICP-MS for the metals, the common metals that are analyzed pretty much everywhere: lead, cadmium, mercury, arsenic. But some other states in the U.S. have more, antimony, and some of these others. And so we basically, with our ICP-MS 7800 ICP-MS system, analyze for the big 4, so to speak, but lots of others, 25 elements in a 3-minute test. The nice thing about this is pretty much every sample you can think of, unlike for potency that has a different sample prep procedure for almost every product, and pesticides, same thing, and most of these other applications, they require very specific sample prep procedures for each product. ICP-MS uses microwave digestion. There's a very efficient way to take pretty much any cannabis or cannabinoid product, digest it, and get it ready for aspiration into the ICP-MS for analysis. So it's very nice, very fast, very efficient, and is what an analyzer, a true Agilent analyzer, meaning that it's a product that went through our, what we call, new product introduction, quality control, QA/QC type process. This is, a true Agilent analyzer, a true product built for this application, and it comes with lots of things when you get the analyzer. Of course, the proven ICP-MS. It comes with the high density, the high matrix interface that's required because this is still pretty high, heavy matrix. The solution created by our experts. It reduces training costs. You're up and running in days. Essentially, it gets installed and you're trained and you're up and running. And the everything you need to get started with, including the pre-configured instrument, the application module, ICP Go software, which is software very specific for the application, comprehensive implementation and assistance, so get you up and running, pre-configured method templates, lots and lots of documentation. So it is something, it basically makes elemental analysis very easy to do. Control your whole run with this ICP. So you can see here on a tablet where you're controlling your run, and you can get your results from day one, anywhere. You can be anywhere, and it's compatible with the 7700, 7800 RACP-MS systems. So that's ICP-MS, and again, very, very powerful, very popular instrument for us. So in summary, Agilent offers the complete portfolio for cannabis testing. All of the regulated tests, potency, pesticides, heavy metals, terpenes, residual solvents, microbial screening. We have all of the tools. We have the consumables, and we have cannabis kits designed specifically for this, for the fully developed workflows, very, very detailed application notes, fully vetted applications on all of these platforms, HPLC, LC triple quads, GC triple quads, etc. Mass Hunter software mostly, of course, Open Lab software is part of the potency application, exceptional customer service and support, and eMethods. And with that, I'm going to turn it over to my colleague, Tim. Thank you.
Great. Thanks, Anthony. So at the beginning, Anthony mentioned that Agilent's goal is to deliver complete workflows for cannabis analysis. And one of the new products we've introduced to facilitate those complete workflows are chemical standards. So Agilent now has a facility in Rhode Island that can deliver chemical standards, both organic and inorganic reference standards, as well as certified reference materials for several different markets. These are all ISO certified. Most products are in stock and can be delivered in just a couple business days. And there's also an ability for custom standards to be created and delivered. So for these standards, these include instrument and column calibration, as well as system suitability standards. There are tuning mixes for all of the mass specs, method-specific standards, linearity kits, QC, as well as solvents, reagents, and buffers. So for the cannabis methods that Anthony has been discussing, we have a host of standards for cannabinoid analysis. These are not yet available, but they will be coming soon. You can see in the list at the bottom, there's a mixture. We have mixtures for 4 different cannabinoids that contain approximately 2 to 3 standards per mixture. These are available at 1 mg per mil. And so if you purchase these, they can be combined together, giving you a standard at 250 micrograms per mil. And you can use this to dilute to create your calibration curve, get up and running very quickly. We also have 11 individual standards that can be purchased, also at 1 milligram per mil. And all of these standards are only available in the US and Canada. So for the other methods, we have several standards for pesticide analysis, and currently these cover the pesticide list for California, Nevada, Oregon, and Colorado. We will be adding additional state-specific mixes and updating the existing ones to meet the 2020 guidelines soon. you can see we also have several different mixtures of standards for residual solvents for the GC-MS testing, as well as several mixtures for terpene testing. And so all of these can be purchased through our website. So next I want to talk about another new product that Agilent has introduced, and these are our cannabis kits. So currently we have cannabis kits for cannabis and hemp potency, as well as pesticide and mycotoxin kits. So it can be overwhelming with ever-changing requirements for cannabis potency and safety testing. And in order to minimize the need for method development, Agilent has developed and compiled these kits. And these kits include sample preparation productions, analytical columns, as well as supplies that are needed to identify and quantify cannabinoids and also residual solvents, residual amounts of pesticides and mycotoxins. So with these kits, you can maximize your productivity and uptime. They allow for one-stop shopping. One part number gives you all of the supplies and consumables needed to get you up and running and start testing. And because these kits were based off of the application notes that Anthony just covered, they allow for easy method development. So what's in each of these kits that we have? At the moment, we have 4 available with 1 more coming soon. So the cannabis and potency kit will be coming soon. And you can see that we have sample preparation supplies. So these are going to include your SPE cartridges and filters, sample containment, so vials and caps, as well as LC supplies, which will include mobile phase modifiers. We also include, depending on the kit, either an LC column and GC column, as well as guard columns that are used with these. So these kits are designed to allow for 400 cannabis samples or hemp samples per kit. And you can see that we have a kit for cannabis and pesticide mycotoxin by LC-MS only. And then we have 3 different kits for LC and GC combined. So as Anthony mentioned, some pesticides require GC-MS analysis. So with the kits that have the combined LC-MS and GC-MS, you can test all pesticides over all the lists. And the kits that we have for these are region specific, so and also platform specific. So we have 1 kit designed for the GC-MS Intuvo, and then we have 2 different kits designed for the 7890 and the 8890, 1 for the combined US list and 1 for the Canadian list. So beyond just the supplies and consumables that come with these kits, there are also application compendiums that outline the potency and safety kits and provide you with sample preparation protocols, as well as instrument parameters that help you start running samples quickly and efficiently. And they also include user guides, which are kit specific, to walk you through each component included in your kit. And these user guides also include tips and tricks specific to the kit. So Agilent is a single source for your entire workflow. We have cannabis consumable kits, chemical standards, instrumentation, software and reporting tools, and Agilent also provides service and technical support. So from end to end, Agilent is able to assist you at any step of your cannabis analysis. So next, I'd like to talk about a new product Agilent has introduced called eMethods for the cannabis and hemp market. And these are designed to be an end-to-end workflow program to quickly get you up and running and assist you and help take care of some of the method development needs that would normally be required to set up a new method. So typically the steps to take before analyzing cannabis require a lot of work. You have to research the analysis, understand the requirements, choose the right instrument and consumables, verify your sample preparation, optimize the analytical methods, and streamline data analysis and reporting. And this can take anywhere from weeks to months to get from the beginning until you're ready to start analyzing samples. So Agilent has done the hard work for you. We have application notes that we've developed and taken care of all of these steps listed. So rather than starting at the bottom of the hill, with an eMethod, you start 80% of the way, and you can get up and running much faster. So what is contained in an eMethod? So we have multiple eMethods specific to applications. They contain extensive support information, as well as analytical methods. And so these are designed for Mass Center and Open Lab, and the information and methods are packaged together into an eMethod. And this eMethod can be distributed and installed on PCs, and we have eMethods available for all of the key applications that Anthony covered. So there are multiple components to our eMethods. All of them are electronically delivered. They can be downloaded from our SubscribeNet website that we use for software delivery. And so they contain information about the right instrument and configuration, optimized analytical methods for sample introduction, chromatographic separation, detection, and data analysis. They contain a list of expertly selected sample preparation protocols and consumables, best practices for sound implementation, as well as the application notes that these are based on. So this is what our eMethod software looks like. When you load the software, this window comes up and you can see that there is an acquisition method, a quantitation method, an example data file, and then an additional data file. And that location where those additional data files can go can be user-defined. For the methods and the data files, these are designed to install into the appropriate Mass Hunter folders so that they can be immediately loaded into the software and used. So there's also support information included with these eMethods. You can see there's instrument configuration. We list our software compatibility, give background information, list the supporting files that are included. We outline sample preparation and include this, and we also include a list of consumables, including part numbers, to make it easy to find everything needed to get up and running. These Agilent eMethods, as I've mentioned, the goal is to shorten startup time and maximize profitability. So with a DIY approach, there's a lot of steps that are required from when you want to get started to when you can actually start analyzing samples and generating revenue. Using Agilent eMethods, we've shortened that time from startup to analysis. And this, we've also put in the time to verify these methods and ensure that they work properly. So using eMethods, you're able to get up and running faster, which can be a significant cost saving. So how will these benefit your laboratory? So as I've mentioned, Agilent has done the work for you. We've done the initial method development steps and packaged all of this information together, which can be quickly installed and used. So this minimizes risk and effort into the design and planning of the analysis. It eliminates delays. There's no missing or incompatible components. It can maximize profitability by allowing for quick startup time, and it increases lab productivity by using efficient methods. So finally, I'd just like to wrap up. We have safety and quality testing necessitates a suite of analytical instruments, and Anthony covered the different instruments required. Agilent offers a portfolio of instruments to cover all cannabis quality and safety testing. Our applications and methods have been developed and rigorously tested to meet all regulatory needs. Our Agilent Rhode Island site has a large inventory of chemical standards and reagents that are needed to meet cannabis lab requirements. We now offer consumable kits, which bundle all consumables needed in an easy-to-order single part number. And we now offer a suite of eMethods for fast and easy implementation in your lab. So I'd like to thank you for attending, and now we'll have a Q&A session.
And thank you, Anthony and Tim, for that informative presentation. And we will now start the Q&A portion of the webinar. Just as a reminder, if you have any questions you'd like to ask, please do so now. Just click on the Ask Question box located on the far left of your screen. So let's get started. We have quite a few questions already coming in. The first question is for you, Anthony. Why is the terpenes analysis done liquid injection instead of Headspace?
Okay, thank you. Well, you kind of look at the history of this market. Initially, the approach was to use Headspace for the terpene analysis, and you would see that, pretty much across the industry. But as time grew and, the market matured, many laboratories realized that there was some problems with Headspace analysis of terpenes, especially in the presence of, high cannabinoid content products like we're seeing today. And one of those is the loss of higher boiling, heavier sesquiterpenes like alpha bisabolol and others that you begin to lose, as I mentioned, in these heavy matrix, high cannabinoid products. And you can't dilute. Now remember, terpenes in plant material tend to be about 1 to 3% by weight. So that's 10 milligrams per gram, 30 milligrams per gram. That's very, very concentrated. But in some of these products, you may have much, much higher content, 22%, 30% of certain terpenes. In fact, in our recent liquid injection application note that we just completed, just published last week, we looked at one of these products and it was 22, 30%. So you need to be able to dilute that down, which you can't do with Headspace. And lastly, terpenes analysis has very traditionally been done in the food and flavor industry by liquid injection for many, many, many years. And so with the market trending towards liquid injection, we felt that the time was right to get out there, get out in front of that, and get that application for our customers.
Thank you, Anthony. Our next question is for Tim. How can customers access the eMethods?
Great, thanks. So customers will be able to access the eMethod once they've been purchased on our SubscribeNet website, which Agilent already uses for our other software delivery. Customers will receive a registration code. They can sign in and create an account, and then they'll be able to download the eMethod directly to their computer.
Thank you so much. And again, thank you, audience, for the great questions coming in. We have time for a couple more. Our next question is for either one of you. Are the cannabis potency kit with the preparation protocols matrix specific? Example, flower or extract?
Yeah, I'll take that one. So there are protocols that are provided in the application notes. Now, we've divided the application note into cannabis specific or hemp specific, and that's because of the laws in the U.S., but the application itself and the preparation are the same, and it is for flower or inflorescence, oils, resins, tinctures, and concentrates. So we provide, some procedures for those particular matrices. There's many others. There's certain edibles, there's gummies, and you'll see more of that different matrix work come out as we move forward. Gummies in particular, we found that you bring them up in water, sonicate, and they melt into the solution, and you can take it from there. So that is the challenge with many of these applications, is the sample prep is very specific to that particular product. And really only with microwave digestion do you see that kind of narrowed down to where you do pretty much any product for ICP-MS and microwave digestion.
Thank you. Our next question is about the systems. Are all of these systems 21 CFR Part 11 compliant?
So we have compliance features that can be turned on in the software. Most laboratories that are in the laboratories themselves are ISO 17025 compliant, and we have the ability to, within our software, turn on compliance features, audit trailing, those types of security issues, user levels, super user versus a standard user, so to speak. So yeah, we have that particular parts that can be turned on in the software. I will say that many of the labs so far, they're just now, the market's just now, I think, starting to think about this part of it as it matures. But yeah, we have those features.
Thank you, and thank you again, both of you. We have one more question. Who and how create the eMethods at Agilent?
Okay, I can take that one again.
Go ahead, Anthony.
We'll both take this on. I'll go first. It is a team effort, and it begins with the application. Many of these applications have been developed with my colleagues, Tim and others, in the field, building these applications and then bringing them in-house to finish them off, qualify them, vet them, put them through the torture tests, as I mentioned earlier, and then get it out there. And then we work with our other colleagues within the organization to develop the eMethod tools, the software tools that go along with that. And of course, we're working with our chemistries and supplies division to integrate their products, so it really becomes this kind of triangle of application, eMethod, and consumables all working together.
And Tim, did you want to add to that?
No, I think that's a great answer.
Okay, perfect. Anthony, Tim, thank you very much for this presentation. Did either one of you want to provide some closing remarks before we close today?
I'd just like to say thank you to the audience for joining us today, and feel free to follow up with us with questions. If you do, reach out to me in particular via email, and if I don't get back to you really quickly, just ping me again. I get about 200 emails a day, so sometimes it's hard to keep up with them all. But I'm very happy to answer questions after the fact.
I want to thank you both
as well.
Oh, go ahead, Tim. Go ahead.
I was just going to say thanks to the audience as well.
And we want to thank you both again for your time today and for your important research. And before we go, I want to thank the audience for joining us today and for their interesting questions. And remind you that questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand through June 2021. Until next time, stay safe. Take care. Bye-bye.